Hello, good afternoon everybody. Thank you for coming. It's my pleasure to present Amin GHABRIAL, or Amin GHABRIAL, who is from the University of Pennsylvania and has been working on tracheal function and tracheal development in Drosophila. He did his PhD with at Princeton. Suppose working on Genesis, right? Not tricky. But then moved to Mark Krasner's lab at Stanford, where he got into the tracheal system. And he will tell us about how these beautiful structures are formed and how they work in the seminar. So, please.
Thank you. Thanks for hosting me and giving me the opportunity to share some stories with you. So my lab works on Drosophila tracheal system. This is a picture of my favorite cell in the tracheal system. It's called a germinal cell. So a single cell, that's the nucleus in red, that makes dozens of branched cellular extensions, and each one of those cellular extensions has a tube running through the middle. I'll tell you a lot more about this later.
So this is one cell. The whole green thing is one cell.
Right, right. So just for perspective, this is probably covering an area of 400 square miles. So another level of things are quite large cell. Okay. So in general, we're interested in how cells make tubes of the proper size, shape, and cellular architecture. And so I guess you guys are mostly Drosophilists in the audience. I wasn't sure how many outside folks we have, but just for sake of comparison, this is human lungs, which is a branched tubular organ composed of thousands of tubes of different architectures, sizes, and shapes. And this is the human vascular tree, similarly complex branched tubular organ. And together, they accomplish essentially the same function. Oops. Same function as the Drosophila tracheal system, which is much simpler network of branches. So whereas these have millions of cells, I keep hitting the arrow.
If it bothers you, we can take the.
Let's see if I can.
Yeah. Or we can take the transponder out, and you can only use.
There. We'll see. So this branch network of tubes is composed of about 1,600 cells. So much simpler, and those cells are all arranged in a simple epithelial model. And within that branched network of tubes, there are really three different tube architectures. So the largest tubes are called the dorsal trunks, and they consist of 2 to 5 cells around the circumference of the cell. And these are polarized epithelial cells, so they're apical domain lines the luminal surface of the tubes, and basolateral faces. There are also single cell tubes. These that we call autocellular tubes are wrapped around the long axis and make self-adhesions to seal into the tube. And then there are 2 cells at the tips of the branches. One called a fusion cell that makes an anastomoses to the contralateral branch, so it connects the network. And one called the terminal cell that I showed you a picture of at the very beginning slide that makes dozens of branches, each of them containing a seamless tube. And here is just an EM section of one of these terminal cells. You can see the tube in the middle of the cell with no connection to the basolateral. So this is a true intracellular tube that's in cross-section seamless. Whereas if you were looking at a cross-section of these tubes, you would see that autocellular junction as a seam that runs the length. So since there's so few of us, please interrupt me if there's anything that's...
I promise to do that. So the dorsal trunks are the main, when you see the whole animal, are only the long main...
Right. So
these
are the dorsal trunks that run from the tail to the head.
And everything else is autocellular or terminal.
Most of the others are autocellular or terminal. And I was showing you kind of a blown-up view of one of these branches that we call So before we get into the stories, I just wanted to draw some parallels to the vascular system. So one thing is that the branches of the tracheal system migrate out from what's originally a sac of cells, epithelial cells that have been vaginated from the ectoderm. And those cells, during the migration, are guided by two cells called tip cells that migrate up an FGF gradient. And this is very similar to what happens during sprouting angiogenesis in the vascular system, but with the VEGF and VEGF receptor rather than FGF. And last, the tip cells that in Drosophila are going on to become fusion cells or terminal cells and make seamless tubes. In the vertebrate vascular system also make seamless, what are called seamless epithelial cells. So seamless tubes in the vascular system. And this is 3 seamless tubes in the zebrafish vascular system. So the approach we've taken is a classic forward genetic approach, but the screen we used was a genetic mosaic screen. So it's a little bit dark, but hopefully you can see all of the tracheal system is labeled in red, and then we mark our homozygous mutant cells positively with G. So I screened the 3rd chromosome, which you guys all know corresponds to about 40% of the Drosophila genome. And we identified about 75 different complementation groups that gave 2 morphogenesis defects. Most of those affecting the seamless tubes, which is why we've gone on to focus mostly on seamless tubes. These are just some that we haven't reported on yet. I don't know if you can see the gene names, but hopefully you can. And then there are 10 additional mutants that we've been able to publish on so far. And today I'm going to tell you 3 kind of interrelated stories about projects on mutants from the screen. So one mutant was called braided, which turns out to encode the Drosophila syntax in 7, also called avalanche. And so these mutants have defects in early endosome, and they, oops, did it again. They show an expanded apical domain. The second story I'll tell you about is characterization of a mutant called Wheezy, which encodes the only Drosophila germinal center kinase 3 submember. And these mutants also have an extended apical domain. And the last story will be characterization of 2 mutants, Ogal and conjoined, that encode 2 subunits of the vacuolar ATPase. And these mutations cause a reduction in size of the apical domain. So as you can tell, all 3 of these stories converge on the apical membrane domain, which in retrospect makes sense because it's the, oh wow.
That was the last bit of light. That's the best we can do. The rest is all coming from the sun.
Yeah, I'm sorry.
It's also the projector is not your fault.
Connector, and so we're operating from a PC. I'm not sure how to adjust the brightness or contrast.
Does anyone know how to adjust the brightness of this, perhaps? Anyone want to venture it?
Well, for those of you with keen eyesight, you might see red labeled here, which is an antibody that serendipitously labeled the apical membrane or the luminal membrane of the seamless tubes. And here you see foci of crumbs. And in the merge, you can see that these foci of crumbs mark the apical membrane, or the luminal membrane, meaning that
because there's no apical, right? That in the seamless, you don't define apical, right? It's
so
the lumen.
So the luminal membrane is apical, is the point I was trying to make. So this is generally true in all epithelial tubes, that the luminal membrane is apical, and that's also true in these seamless tubes. So in retrospect, it's not surprising that a lot of the mutants that we find have seamless tube defects or affecting the size of the apical domain.
Change the way.
Can I ask something, because perhaps I missed it? You said that the 2 tip cells make a seamless tube, or 2 seamless tubes, or what?
Yes.
I was confused.
Sorry, they go on to differentiate as a terminal cell and a fusion cell. And the terminal cells and the fusion cells both make seamless tubes.
Okay, now it's.
Better?
Yeah, much better. Great.
Sorry, so this antibody is a specific protein that recognizes from that membrane.
Yeah, so what I was saying is that we came upon this antibody kind of by good fortune. It was actually raised against a protein that we are working on in the lab as a peptide antiserum. That it still stains the seamless tubes in a normal. So it recognizes something else. We don't know the identity of that, but it's quite useful for us.
And it's specific only for the seamless tubes, not for the lumen in general?
It does stain the larger tubes, but not nearly as well.
Right. Okay.
So the first story I was going to tell you about is this braided mutant, which turned out to encode syntactin 7. This is just a wild type control clone. You can see the terminal cell has dozens of branches, all of them filled with a seamless tube. So the cytoplasm is marked in white and the seamless tube in magenta. And in the braided mutants, you can see a big reduction in the number of branches, but also a change in the morphology of the seamless tubes. So they have these little dilations all along. So these are just shoal diagrams showing and giving you an idea of the complexity of the branching in the cells. So wild-type terminal cells, if you draw circles of different diameters, 50, 100, 150 so far, so on microns from the center of the cell, you would intersect this number of branches. The y-axis isn't showing up, I'm afraid, sorry, but this is an increasing number.
It says number of branch intersections. The numbers are those.
Yeah,
okay,
the y-axis for some reason doesn't have the actual numbers. Yes, but in braided mutants, you can see that we still get long branches, but there's a reduction. Okay. So the gene affected is, the Drosophila syntactin 7. And we had two mutant alleles, Q6 and 1615, that were nonsense mutations. So they're truncating the protein and are most likely null mutations, functionally null. The other thing I wanted to point out is that syntactin 7 has previously been worked on by David Builder's group, who also identified ribosome 5 and DPS 45 as cooperating with syntactin 7 in SRAB5 dependent early endosome pathway. And we see similar defects if we look at mutations in any of these components. GHABRIAL, or the Drosophila dynamo, we can also get related phenotypes, but we have to kind of titrate the knockdown, because if we look at null mutations, we get a much more severe phenotype.
And so it's a V snare for the early vesicle fusion with the early endosome.
So in wild type trachea, you see RAB5 staining these large foci. In braided mutants, you see RAB5 being very dispersed in non-accumulating mutants, normal looking enzymes. Okay, so next we wanted to understand at the cellular level why we're getting these dilations in the tube. And one of the first things we noticed is that we're seeing a great accumulation of crumbs and phosphomories in the apical membrane. So these are antibody stains in a filleted larvae. And here is a wild type heterozygous cell, because remember we're looking at this in mosaic animals. So here you can see the apical membrane and those little foci of crumbs that are labeling it. In contrast to within the mutant clone, where you see dilated apical membrane and a much stronger accumulation of crumbs.
So really the dilation is amazing. The width of the lumen is visible in the mutant, it's invisible in the wild type, right? Is it visible if you up the magnification, the wild type?
Yes.
You do see it.
You can see it. So it depends on where you are. If you go out to the tips, then it looks more like a single line. If you're closer to the
So it could be submicron then.
Yes.
The wild type could be it gets down to submicron.
Yes. So here we're looking at a couple of microns in diameter, and out of the tips, less than But you're absolutely right that these cells have wider diameter lumen. Even in the soma, it's maybe twice as wide as well. And then here we're looking at accumulation of phosphomolysin in a heterozygous cell compared to a neighboring wild-type cell. Again, you can see a pretty big decrease in level. It also turns out that if you overexpress wild-type crumbs or a phosphomimetic form of molysin, that you get a phenocopy. So you start to see dilations in the tubes, fewer branches and larger diameter. So in these mutants, we're really not affecting endocytosis, but just the accumulation of these proteins on the apical membrane, and that by itself is sufficient to give us these two dilations. It's also a phenotype that we can modify by reducing the levels of molysin. So here's the wild-type terminal cell, a braided mutant, and a braided mutant in which there's only one wild-type copy of molysin. You can see a strong rescue of the phenotype. So here we're just quantifying the number of cysts per millimeter of the tube. So you can see in the mutant, quite a high number, and about half as many dilations in cells with half the amount of molysin. And what
do you mean by number of cysts? How do you define it? Sorry.
Right. So just counting the number of these dilations.
Oh, the super big dilations, right?
And then to get an idea of how much seamless tube is made, scoring the total length of the tubes as a proxy for measuring branching. And we get this rescue without restoring the normal pattern of protein receptors. So the ratified is still disturbs.
And like overexpressed as well, over accumulated, right?
So lots of little discrete ratified vessels. So at this point, I wanted to just pause and say that although crumbs is essential very early for all epithelium, if we make cells that are, if we make animals that are mosaic for crumbs, so zygotic loss of crumbs, we see no defects in the tracheal system. So in that context, it's interesting that the cells are so sensitive to increased levels of crumbs, but not so sensitive after this early embryonic requirement to reduction in levels of crumbs. So here we're doing an endostasis experiment, looking at a cell that's doubly mutant for braided, this is taxon 7, and crumbs. And what you can see, hopefully, is that compared to the neighboring heterozygous cell, the double mutant no longer has that highly elevated level of phosphomolysin. So we interpret this as crumbs acting to recruit phosphomolysin to the apical membrane, which is consistent with data from other groups showing that crumbs and phosphomolysin physically interact. So what's going on? What we think is happening is that it's really the levels of phosphomolysin that are critical, and the actin bundling effect of molysin. So if we look at actin enrichment at the apical membrane, we see, we see a great enrichment in the intensity, looking either at with actin GFP or life act Ruby, both in the braided mutant, crumbs overexpression background, or the phosphomimetic molysin.
Remind us, life act has a
So
special
life
feature.
act,
It has.
Yeah, it binds to actin.
Right.
So actin GFP has some gain of function phenotypes, so it's maybe not the best marker to use to follow actin. So we also repeated this with life act.
Which is non-lock signal.
Right. And the last point I wanted to make is that if we make clones that are mutant for twin star, which is the Drosophila cofilament, so an actin 7 protein, we get a phenocopy where we get these dilations in the apical membrane. So this early endocytosis story really led us to looking at regulation of crumbs and Louisin in the apical membrane, where they are playing a critical role in cortical tension by controlling funneling of actin. And why exactly that results in these dilations in the tube is the next question. If anyone has some insights into that, I'd love to hear.
Myers. Yeah, actually, it was Myers.
So that's a good question. Marcus Folter in his lab has been trying to look at that, and so far they haven't found a myosin environment.
How many myosin genes in flight? Several, right?
Yeah.
Okay.
So now we're going to move on to the next story. Here we're going to be looking at the connection of seamed and seamless tubes within the terminal cell. So this is just a schematic. Here would be an autocellular tube. Here the intercellular junction between that autocellular stalk cell tube with the neighboring terminal cell. And a number of years ago, the Krasnow lab showed that these terminal cells usually have a little bit of an autocellular junction between the intercellular junction and the terminal cell nucleus, and that you then transition to a seamless tube. So we wanted to identify mutants that might disrupt that connection. So we looked for mutants that had defects specifically in this region that we're calling the transition zone. We were transitioning from a seam to a seamless tube. Here's just a micrograph of that same junction that I had a cartoon of. So here's the intercellular junction using cortical to label the septate junctions, equivalent to the tight junction invertebrates. But you'll see the same thing if you use EK instead. So the autocellular junction, the intercellular junction, and then autocellular junction within the green labeled terminal cell. And you see something kind of similar if you look at the zebrafish vascular system. So here is a seamless epithelial cell with a red blood cell passing through it. This is the intercellular junction, and what looks like a little bit of an autocellular junction. Okay, so one of the mutants that we found with one of these local defects between the intercellular junction and the terminal cell nucleus was a mutant we called Wheezy. And they showed these large dilations very close to the intercellular junction, but within the terminal cell. Wheezy turned out to be a mutation in the only Drosophila germinal center kinase 3 family member. So it's a sterile 20 kinase superfamily. Humans have 3 of these that turn out to be functionally redundant. So Drosophila is a useful model system in that way.
Are these are implicated in something particular? Or,
So that's a good question. The long-standing thought in the field had been that they were part of the MAP kinase cascade, but people are now kind of backing away from that. And the million-dollar question is, what is the target? Which we're hoping to get at. So since we had a single allele, we wanted to be careful and make sure we really identified the right gene, especially since we were doing mosaic analysis and we're looking at cells that are homozygous for an entire chromosome R. So we can phenocopy with RNAi. We can rescue with a wild-type cDNA transgene. And we also showed that the kinase dead version of the gene doesn't rescue. And in fact, it acts as a dominant negative. So if you overexpress the kinase dead, you generate a kind of mild dilation. Okay. So the reason that this was really interesting to us is because these proteins are known to bind to a human protein called cerebral cavernous malformation 3, which is affected in human vascular disease. So some people you may have heard of, Florence Griffith Joyner and Alberto Contador, are both famous people who have suffered from this type of disease. Florence Griffith Joyner actually passed away from such a lesion. Alberto Contador had brain surgery to remove one of those dilations in the vascular, cerebral vascular system.
Aneurysms and things like that.
Yeah, it's a large, leaky tube, capillary around the brain. And he actually came back to win the Tour de France afterwards, although, as we all know now, with a little bit of extra help. But I guess everyone was having those extra edge back in those days. So it turns out to be a surprisingly common disease. About 1 in 200 in the general population are affected. So the cerebral capillaries, but not capillaries elsewhere in the vascular system, become grossly dilated. They lack support cells, parasites, and they become leaky. So this can give rise to a range of defects. Turns out 25% of patients that have these lesions are actually asymptomatic. And then some have mild symptoms like headaches, but it can go all the way to stroke. So it can be quite serious. And there are familial as well as spontaneous forms of the disease. And the familial forms, they used to say 70 to 80% of familial forms were linked to mutations in one of 3 genes. People now seem to think that, in fact, all familial forms can be associated with mutations in one of these 3 genes. CCM3 is the only one of those proteins that's conserved in FOM, but there's a nicely conserved ortholog.
Also, between them, they're not parallels?
They are not parallels.
They're
so
different.
Even though they have CCM names, they're completely unrelated proteins, although they do form a ternary complex in vertebrates that have it. So CCM3 is probably the most ancient member of that complex. And the portion of the protein that's critical for binding to germinal center kinases is conserved, and another group has shown that the fly orthologs do physically interact. So we knocked CCM3 down by RNAi, and we get a mild dilation defect that's specific for the germinal cells. It wasn't fully penetrant, so we went ahead and made a knockout by what's now old school technology, not using CRISPRs, but autologous recombination. And we found that when we did our mosaic analysis, we saw 50% roughly 50% penetrant defect. And that turned out to be due to maternal deposition. So if we make germline clones to remove maternal CCM3, and in that context, in that background, then make zygotic clones, those cells show 100% penetrant to dilation defect, identical to the GCK3 defect. We can rescue the CCM3 mutant phenotype with a wild-type cDNA, as you would expect, but we can also get pretty good suppression of the CCM3 mutant phenotype by overexpressing a wild-type form of the germinal center kinase, so we can bypass the requirement for CCM3. Okay, so at this point, I just wanted to pause and say that our system generating mosaics is, to some extent, modeling the human disease. So the human disease is autosomal dominant, and the idea is that you're born with one mutant copy, you spontaneously lose the second wild-type copy, and that gives you a local lesion in the vascular system. So we're more or less doing that by inducing autologous recombination in our mosaic animals. And the other thing I wanted to emphasize is that when we make clones, we see defects only in the terminal cells, not in any other cells in the tracheal system. And that's true even when we do pantracheal knockdown with our RNAi. So it's really these specific cells that are being affected, and that makes us speculate that maybe the vascular tip cells could be the cell of origin of the disease. So now on to kind of a cellular characterization of these defects. What's really going wrong in the transition zone of these cells that lack CCM3 or GCK2 function? So here I'm showing you the seam of an autocellular tube, the intercellular junction, and then the terminal cell that's making mostly seamless tube. I guess one thing I should point out is that in most third instar larvae, that little bit of autocellular tube has been remodeled away. So when we look at third instar, it's only 20% of the cells that still have that autocellular tube remaining. 80% seem to have modeled away and have only seamless tube. In contrast, in the GCK3 mutants, we see a perdurance of that bit of seam tube. In addition, when we look at components of septate junctions, we see inappropriate localization out towards the tips of the cells in places where you normally wouldn't expect to see septate junctions. So back to our old friends, phospholamine problems. So this is a fillet of a mosaic larvae. Our homozygous root cell is labeled with GFP.
Third dister, right? Third dister.
And phospholamine in red. So I think even in the front row, you probably, at this exposure level, you don't really see the phospholamine in the heterozygous cell. In contrast, in the homozygous beta cell, you see a strong enrichment all along the apical membrane, and especially right around that local dilation at the base of the joints. If we look at crumbs, again in wild type, the dots outline the terminal cell, and you can see these little foci that decorate the luminal membrane. In these GCK2 mutant cells, you see a strong accumulation of crumbs. As I showed you in the first story, overexpression of crumbs or phospholamine is sufficient to give you little microdilations throughout the cilious tooth. We see that in CCM3 and GCK2 mutants as well. But what you don't see is that large dilation between the intercellular junction and the terminal cell nucleus. So it's recapitulating part of the phenotype, but not the entire. Nevertheless, it seems that overexpression of crumbs is essential for producing these local dilations, the large local dilation phase, because, if we knock crumbs down by RNAi, we shift from seeing about 80% large dilations and 20% small dilations to getting 60% of the cells having no visible dilation. So strong suppression. Likewise, we can suppress the large dilation defect by knocking down varicose, which is a component of the septate junction. Again, you get a pretty strong suppression. Below in these backgrounds, we see a new category, which we're calling, the gap category. So here you can see the seamless tube within the terminal cell. Below here, if I was showing you, you would be able to see an air-filled tube in the autocellular stalk cell. But there's a gap where you see positively related cytoplasm that doesn't have a tube. So the seamed and seamless tubes in this category are not connected anymore. Okay. So as I mentioned before, the million-dollar question has been what's downstream in this pathway. We don't know for certain yet, but we have some clues that I'll share with you. Our first clue came when we were looking at RAB11. This is RAB11 staining in a wild type background. Unfortunately, we've also labeled the nuclei with GS red, so that large red blob is not RAB11, but the nucleus. These little small red foci are RAB11 staining, so RAB11 marking the recycling enzymes. In GCK3 mutants, again, we see the nucleus, unfortunately, but what's really striking is this enrichment of RAB11 right around the side of the tube dilation. So this result we had with an antibody generated by another lab that we could get more of. So we went on to try to characterize it further using YFP tagged RAB11, but no longer obviously looking at endogenous RAB11, but overexpressed RAB11. So when we do this with just a wild type YFP tagged RAB11, RAB11 looks like it's everywhere now, no longer restricted to those little foci of recycling endosomes.
You don't see, you're not imaging it.
We are imaging, so the green is the YFP.
Okay.
But because we're overexpressing, you no longer see that beautiful.
Yeah.
And same thing is true in the Wheezy mutant background. So red is labeling the luminal membrane, so you see that dilation. but this turned out not to be useful. The same was true with the dominant negative form of RAB11, but the constitutively active form of RAB11 turned out to be useful. So ignore the nuclear staining that you're seeing here. That's, again, a background problem. But the cytoplasmic staining is the constitutively active form of YFP tagged RAB11, and it's throughout the terminal branches. In the GCK3 mutant, we've switched labels, so now YFP is in red. You see this massive localization around that large dilation, and then RAB11 spreading along the apical membrane. So we've confirmed our staining against the endogenous RAB11 with this transgene. And interestingly, it seems like the effect is specific for the active form of RAB11, so the GP locked form. This is functionally meaningful, so if we make GCK3 or CCM3 clones and express a dominant negative form of RAB11, we suppress the tube dilation defect. And we can also use classic loss of function wheels in RAB11 and get very strong suppression of the gene.
Control RAB11 downregulation alone does not
Causes other problems.
Okay. Which are rescued by the Wheezy clone?
Which seem to be kind of
counterbalanced by the.
So, sorry, this is just showing the same thing, but in the CCM3 background instead of the GCK3. Okay, so how do we put this all together? We think, just as in humans, that CCM3 and GCK3 are working together, but that they're acting to restrict RAB11-dependent recycling. So in our mutant cells, we see disruption of septate junction remodeling. We get perdurance of that autocellular junction. We think because of the inappropriate recycling of those septate junction components back to the plasma membrane. We also see inappropriate recycling of chromes and phosphomucin, getting that accumulation of high levels at the apical membrane. And we think that the targeting of those RAB11 positive vesicles is guided towards the septate junction, which is acting as kind of a signpost for transport of those vesicles. And that's why we're getting this large dilation, specifically in this region of the terminal cell, where the septate junction is in close apposition to the gene stream. Any questions on this part? Move on to the fruit story. Two mutations that we have been working on in the lab that we would call conjoined and oak gall. So people recognize conjoined twins. This is an oak gall. Many of you are probably not familiar with it. But these are actually insect-induced outgrowths that you find on oak trees. And we saw corresponding defects in the tracheal system that reminded us of these. So here I'm just showing you an autocellular tube, in both cases GFP labeled. In this case, we're labeling all the tracheal cells with DS red, and here just the tracheal nuclei with DS red. Those are control clones. In conjoined mutants, instead of getting cells rearranging so that they are going from side-by-side configuration to make a multicellular tube, sliding past each other to make autocellular tubes, we see those cells remaining connected to each other to make a multicellular tube. So that defect is what led us to name one mutant conjoined. And here you see the oak gall phenotype, where the cell, instead of being elongated along the long axis of the tube, is kind of rounded up. It doesn't contribute very much to the tube. You can see kind of green just here surrounding the tube.
So the tube is shortened by this way?
It is short.
It's happening this way.
Right. So if you were labeling the neighboring cells with DS red, you would see the cytoplasm in close apposition to the tube. So the cells that make the autocellular tubes are really elongated and not so fat. So So these cells are really kind of bulging out from the rest of that. So despite these different names, these two mutants actually show exactly the same set of defects. Conjoined mutants will also show this defect, and adult mutants will also show the conjoined defect. In fact, just by looking at the mutant cells, you can't tell them. So a graduate student decided to work on both of these. The other thing I'll mention why we started to look at these is because they had a dilation when we made terminal cell clones. Sorry, they had a gap in gas run when we looked at terminal cell clones. So reminiscent of that gap phenotype I mentioned earlier.
So this is the same terminal cell.
Right.
Just the green doesn't show well as represented. It's not monotomers.
We'll get back to autonomy in a minute, because it turns out to be a strong non-autonomous component to this effect.
Well, it makes sense. If one cell pulls out of the,
Yeah.
the neighboring cells have to do something about it, right?
Okay. So we're back to this very familiar cell-cell interaction. So the terminal cells that we're marking in green, now we're ignoring all of the branches and looking just at this portion of the terminal cell closest to the intercellular junction. So here we're looking at the adherence junctions now instead of the septate junctions, so EK-adherence staining. You can see the ring of the intercellular junction, and then the autocellular seam along the stalk cell, and then the terminal cell, no such autocellulars. Okay. In OCO and conjoined mutants, we saw two defects. In one case, a defect we're calling multiple entry phenotype. So a single terminal cell, but now the neighboring stalk cell is branched, and it connects to the terminal cell twice, okay, with two intercellular junctions. And the other defect, here we're looking at the terminal cell, and you might be able to make out is this red line of the adherence junction that's extending all the way out to the tip of that terminal cell. So normally, the seamed tube would stop somewhere around here, and this would all be seamless tube. But now we're seeing a seam that seems to end in a ring. And then we see a little bit of tube that extends beyond that. So OCO and conjoint turned out to encode two different subunits of the vacuolar ATPase, VHA13 and VHA26, which are the G and E subunits. And those two proteins actually form a heterodimer that together is called the stator. And that connects the V1 domain of the vacuolar ATPase to the V0 domain. And the stator regulates the assembly and disassembly of the whole enzyme. So when the enzyme is assembled, it acts as a proton pump, and it acidifies intracellular compartments. When V0 and V1 dissociate from each other, V0 can then have a separate function, interacting with snares to promote membrane fusion. So the first question we had is whether we were seeing a loss of function in the proton pump or a gain of function in that membrane fusion function. So we knock down different V0 subunits, and we get a nice phenocopy. So you can see the adherence junction running all the way along these tubes that should in principle be seamless. So it's not a gain of V0 function, but instead probably a loss of proton pump function. So that's just a quantification. And then to verify that it wasn't a structural role of the whole enzyme, but it's the enzymatic role, we also looked at point mutation in the V0A subunit, R755A mutation that allows assembly of the whole enzyme, but specifically blocks the proton pump function. And that gives us, again, these adherence junctions that are running along what should have been seamless tubes. Okay. So, why is this happening? We thought of a couple of different possibilities. One thing that seemed really potentially interesting is that somehow VHPA's activity was required for the terminal cells to switch to the genesis programs from making autocellular tubes to making a seamless tube. Another possibility that we considered was that maybe the terminal cell normally makes autocellular tubes, and then you would have a membrane-membrane fusion event to convert that to a seamless tube. So maybe we're trapping an intermediate in seamless tube formation. That has precedent in some other systems. For example, in C. elegans, there's the extracorporeal canal system that has a pore, a duct, and extracorporeal canal cell. And two of those cells make seamless tubes. One of them by wrapping and making it self-adherence junction, and then doing a membrane-membrane fusion. Turns out neither of those are correct, and instead, what's happening is that the neighboring wild-type cell is invading the mutant cell. So here, we're using the Flybo system to differentially label adjacent cells, so green the terminal cell and red the stalk cell. And here we're looking in VHPA's mutant background, specifically mutant in the terminal cell. And what you see is that the neighboring wild-type stalk cell is kind of sticking in now to the mutant green cell. So you can see the red membrane sitting, penetrating into that green cell. And the junctions fall. Here's another kind of more extreme example. So here's the green cell. You can see the red cell entering into it, extending this way, extending quite a distance in this direction. Okay. So the neighboring cell is invading. Why? The clue that we decided to pursue was the possibility that these terminal cells just aren't very good at making apical membrane. And the reason for that, we thought, is that if we look at the apical membrane that's actually made by these terminal cells, it doesn't cover this portion of the tube that's actually provided by the neighboring wild-type cell, but it's just this little bit of tube out beyond that intercellular junction.
So this is for reason again? What mark?
Sorry, red is the adherence cadherin.
Cadherin.
And blue is just an autofluorescent component of the luminal matrix. So you can see the tube and the seam. So it's this cell, but we're looking way up here. So a greatly reduced apical domain in these green cells. And that turned out to be true in general in tracheal cells that are mutant for VHPAs. So here we're going to look at a couple of different types of tubes. Here's a multicellular tube. If we make a control filament in the dorsal trunk, you see a cell that's kind of hexagonally shaped. And it's really very thin, so squamous cell. And that red E-cadherin staining more or less outlines the extent of the apical domain. So the surface between these neighboring cells is the apical membrane. In VHPA's mutants, you see a little red circle, which is the E-cadherin connection to neighboring cells. So within that little red circle would be the apical domain of this particular cell. And then cells kind of rounded and bulging out of that field. Okay, so again, greatly reduced size of the apical domain. And the same is going to be true also in the cells that are autocellular tubes. So here's just a control cell. You can see this oval here is the intercellular junction with the neighboring cell. This is the nucleus of the autocellular tube. And then the cytoplasm is stretched thin along the long axis of the cell. In VHPA's mutants, you can see the two intercellular junctions. Most of the cell kind of bulging out from the epithelium, and then a little bit of green that's labeling this short stretch of apical in the right area. Okay, so are there apical polarity defects in BATK's mutants? We looked at crumbs and APKC. This isn't the best picture, but I've showed you several now where crumbs, foci typically decorate the luminal membrane. APKC staining looks much more smooth. You see kind of red all along the apical membrane. That's wild type. In BATK's mutants, you see this massive accumulation of crumbs, foci intracellularly, and the same thing with APKC. We try to figure out what these compartments were. And we see co-localization with trans-Golgi as well as early endosome. So we're getting crumbs and APKC trapped in both of those compartments. If we bypass all of these trapping problems and just knock down APKC or PAR6, so apical polarity determinants, that's sufficient to give rise to these defects where the neighboring wild-type terminal cell is extending autocellular to homozygous fusions or knocked down terminals. So this was something of a surprise. So previously, it had been known that if you had a mutant terminal cell, that neighboring wild-type terminal cells could kind of invade that hypoxic area and supply oxygen to that hypoxic tissue. But this is kind of the first example of an autocellular tube branching, growing, and kind of compensating for a loss of function in terminal cells. So this process of the terminal cell branching and compensating is FGF dependent. The hypoxic tissue is known to secrete an FGF, and the neighboring terminal cell responds by going into that area. So one question we wanted to ask is whether autocellular tube compensation is also FGF dependent, and whether it would then occur under hypoxic conditions. Our preliminary results suggest that that's not the case. FGF doesn't seem to be involved in branching of the autocellular tube, and if we put animals in hypoxia, we don't see a similar invasion of terminal cells by neighboring wild-type autocellular tubes. Instead, we can mimic autocellular tube compensatory branching by physical injury to the terminal cells. I'll just show you that here. So here is a wild-type terminal cell. Again, we're using Flybo to differentially label the membranes of the terminal cell and the neighboring sock cell. And if we injure that cell, so this is pre-injury, and then look 24 hours later, so this is post-injury, you can see that the cyan marked cell has branched and extended into the terminal cell.
So where is the injured part? Close to the, okay.
Sorry, so the way that the injury was done was was pretty crude. Just taking a pair of forceps and pinching. So it's a crush injury.
Just in the junction between the two cells?
Yeah,
or somewhere close?
Around here, near the soma.
Near the soma.
And my graduate students have been able to get similar effects if you reduce apoptosis by expressing KID or gram or revert. And similar to the compensatory cellular hypertrophy described in the ovarian follicle system, either injury or the presence of mutations that cause an undersized apical domain within the terminal cells induces an entry into the endocycle and hypertrophy of the neighbors.
Of the neighboring cell.
Right, so the terminal cell is mutant, then the neighboring cell compensates by entering the endocycle, growing much larger, and branching. That was the last story, and I just wanted to acknowledge the folks who did most of that work. Yanjun Song is a postdoc in the lab who did the CCM3 and GCK3 work with some help from a former technician, Melissa Ng. Jody Schottenfeld-Romes is a former postdoc who did the syntaxing 7 work. And Dina Francis was a graduate student. She actually just defended a couple of weeks ago, who did the VATPAs and my funding sources.
Any further questions?
Jackie,
Yeah.
clones is market in your studies most of
So how did we label clones? It's similar to the Markov principle, but instead, sorry, does everyone know Markov, or should I?
Someone doesn't know.
Okay. So the Markov idea is to make clones in heterozygote animals by inducing mitotic recombination, and then using a GAL80 as a dominant repressor of GAL4 activity, GAL4 driving vertical expression. So in the daughter cells that no longer have GAL80, those are the daughter cells that become homozygous for mutation of interest, and that permits GAL4 to be active and drive expression of the protein. Instead of using GAL80, we used the GFP RNA transgene, but the same principle. When the cell becomes homozygous for mutation, it no longer has the GFP RNA transgene, so we get expression of GFP. The only difference is because we're using GFP RNA transgene, we can label the entire critical system in the case right, which you wouldn't be able to do with Mars.
Continuing with that, did you use tubulin or actin holder or use specific for tracheal system?
So if we were to use Markov clones, we use the tubulin GAL80. We use the breathless GAL4. And for our GFP RNA, it's just the breathless GAL4. Right. Another postdoc in Krasnov made a breathless GAL80, but it actually was to drive GAL80 sufficient enough to work in the Markov system.
So this whole story is revolving around membrane trafficking, RAB5, RAB11,
And we're after the backstory I didn't tell you.
but also vacuolar ATPase also is needed for the acidification of late endosomes. Do they all come together at some point? What do you think? They have something in common?
Yeah, so
I mean, phenotypes are distinct. I mean, the first two are similar. The third one was distinct.
right.
Yeah.
So the three stories are conversion of all of size of the equivalent domain, and the two with increased equivalent domain sort of really seem to come to regulation of phospho no reason. Seems to be where those stories of conversion. The RAB35 story that I didn't tell you about is different.
But also the VA story is,
So,
for example, if it has less apical domain, can you mess around with your other mutations and rescue it, for example?
good question. So we're going to have this kind of
Static, right.
Um,
I'm not sure myself how it would happen here.
yeah,
Simply making a double mutant
So-
of OGOL and GG group, what's it called.
So I think, I'm trying to remember exactly what Dina had done. She had at one point tried overexpressing crumbs, which isn't quite the same. And because there's a trafficking problem, crumbs is going to the wrong place, and that doesn't
Get stuck in the gold years.
So could you overcome the VHP's defect by redirecting traffic in some way? I think the challenge is that we're getting accumulation of crumbs both in the secretory pathway, so the trans-Golgi, and also in the recycling pathway from early endosome. So it may be intellectually harder to figure out what double mutants to make to overcome that problem.
I was thinking simply to make double mutants with your other mutants and see who is more downstream of whom, regardless of what your intellectual, and then build your intellectual model based on the result.
So I don't think we had done that. It's technically a little bit more challenging because all these mutations are on the third chromosome, but we did, I think I glossed over it, that we did now develop a system that allows us to make positively marked clones that are homozygous for 3L and 3R. So it's now a technically doable experiment. We haven't done that.
FORTH-IMBB. 3L, 3R.
Yeah, so those that have existed, but the problem has been positively labeling.
With an end in between. Only when you get double clones, you get positively labeled. Yeah. Any other questions? Yes.
So I'm interested in actin. Do you have any idea what kind of actin filaments? Are they branched? Are they?
Yeah, so that's a good question, and unfortunately our level of resolution is still low. So we know the reasons playing a key role. We know that it's the cortical actin that's right around the apical membrane. But that's kind of the extent of our insight right now.
Okay. Well, thank you again. If there are no more questions,